because of these two persons that invent this kind of Sanger sequencing. This Walter Gilbert and Frederick Sanger then received Nobel Prize in chemistry because of this invention of DNA sequencing. So they did Sanger sequencing, which takes a long time since late 1970. So they took a long time to sequence. And then this is the long sequence, the full sequence. It's a very long sequence taken from a chromosome. Then from chromosome, there is a DNA inside that. The DNA is being sequenced using Sanger sequencing. But then in 2003, 2004, there has been advancement in sequencing technology. We call it next generation sequencing. There's Illumina, there's also Roche, there is also Ion. There are many machineries, also Pacific Bio, PacBio, and the latest one is Nanopore Sequencing. So in this case, instead of getting the whole full sequence, this kind of sequencing technology, they take stretch of DNA. So they take picture kind of thing, like a few stretch, like 100, 150, sometimes 300. So it depends on the advancement of the technology. 300 base pairs, 300 letters in this case. And then there is this nanopore sequencing that has like a newer or longer reads. So, but still, it's not a full sequence. It's still fragments. And this nanopore sequencing can do it like streaming. So while we are pipetting, and then like the liquid is going in, and then the sequence is read in the monitor linked to this sequencer. So sequencer is not this big anymore. It will be this small as a USB stick. And yeah, I think it was in 2015. So this is a typical next generation sequencing data. So it's a stretch. The blue one is the sequence, and the other is just metadata. So this is the header. This is the quality in ASCII number. And yeah, that's what we bioinformaticians use as an input for our process, for our analysis. So this has been the evolution of diagnostic method. We have seen the first one in previous slides. This is called routine microbial diagnostics, and we already moved to sequencing based. So instead of doing this pathogen ID with a biochemistry and physical appearance, we do a whole genome sequencing based diagnostic. So we just sequence it whole genome, and then we can do it faster than the traditional method. And then it's a more ambitious project during my PhD. This is actually my topic in my PhD, where we are not culturing the bacteria anymore. We just sequence it directly from the sample. For example, a blood sample of patient, so we don't need to grow it. We just sequence it right away from DNA samples, of course. And it takes like half or 1 day, but then in reality it's 3 days because people have these breaks and people go home to their families as well. They have life. But then this is even more ambitious project. This nanopore diagnostics, it takes like less than 10 hours to sequence multiple samples at the same time, more than 20 samples. It's called nanopore sequencing, not nanopore diagnostics. So we're going to talk about this. So before I'm going to continue with this metagenomics, I need to introduce you about this culture dependent and culture independent. So in culture dependence, the first two in a previous slide, it was like taking 1 day to 3 months just to grow a bacteria. But with metagenomics, we don't need to do that. It's independent of culture. And then we don't need media anymore. We just sequence it directly from sample. And this is the most important part, the most important benefit of metagenomics, where the organisms that can be cultured are much more like with a culture-based approach. like 1% of the organisms in the world can be cultured. But then with metagenomic, we don't care if it can be cultured or not. We just sequence it, we find it. So all organisms can be detected. And this is like one of the news from, I think it's from one of this journal that they said new techniques has been invented by a team of Danish investigators. This is basically our paper, our first paper in the team that published a new method for diagnosis using metagenomics, which is faster and in great detail, especially important for epidemiologists. So I will talk a little bit about this SARS, this COVID genome. So yeah, as we know already, the genome of a SARS-CoV-2 is like around 29,000 RNA letters, and most of them are ORF protein, non-structural, and then there is a structural protein, which is the most dangerous part, which is the most like changing, most mutated part of the virus, which is the spike protein, which connect to our ACE enzyme in our body, and the other is accessory proteins. So basically, this is the SARS genome, simple, and the solution of this. I mean, to get rid of this COVID genome. I mean, from attacking us, we can just make something that has a strong binding affinity to any of them. I mean, to some part of them, and then so it will what do you call it, like make a binding towards this new drug, for example, or yeah, instead of binding to something that can endanger our life. That's one idea of a drug development, and then yeah, we have seen also some variants of concerns. There are variants of concerns, and there is variants of like investigation. I forgot the other names based on WHO, but I'm just going to talk about this VOC. So it appears in the UK first. There's this alpha variants in the UK, where it spreads faster than the previous one. And then there's this beta, and then there's this gamma, and lately we have this delta variants. And then, of course, oh yeah, this is the other one, variants of interest, sorry. Yeah, there are some variants of interest. This is not of concern yet, or at all, we don't know yet. But this is of interest, the other four. Yeah, and it keeps mutating and mutating because virus, the nature of virus is to mutate. So no wonder it will happen anyway. But yeah, we just need to be prepared for that. And most of these mutation usually they are located in the spike protein part, especially in this delta. They change a lot in the spike, so the spike won't be recognized by some of the current solution, or no, last year's solution. So yeah, so the solution of this all, we already know that some of the countries like the US and Europe, they have kind of solved, but not yet solved the case of COVID, because they are doing a massive and extensive amount of sequencing. So for example, like in Denmark, so everybody has to be tested every 2 days, every 3 days. So for example, a barber shop, they have to get tested by PCR like every 3 days in order for them to be able to cut some customers. Otherwise, they cannot open a barber shop anymore. Something like you cannot cut if you don't show the result of the negative COVID. Something like that. Also, the customer has to show before they're being cut. They have to show the negative proof that of a COVID. So that's why like this massive sequencing. So before sequencing, we will talk about the PCR. So PCR test is like compulsory for everybody. and in order to be active in some places. Otherwise, you should stay home if you don't like PCR test in Denmark. And then after you get tested, if you're positive, then the sample will be sequenced right away by the country, by the government, like sequencing department, something like that. And after that, then we are like kind of detecting where is the mutation. Is it the same one with a Wuhan? Is it the same one with the alpha or something like that? That's why we detect alpha, delta is like very fast, especially when the alpha start attacking like in Europe from UK. Even we know it's from UK. It's because in UK and in Denmark the sequencing is variants called alpha, and then everybody is like from the other country. Even yeah, we have some dramas. It's not only in the Asian countries. In Europe itself, there is drama like Sweden suddenly blocked Denmark for like okay for inventing this COVID alpha variants, but then yeah, like blocking lockdowns, etc. And then finally, like we are now open, but then uh, between I mean during this time, there's also suspicions that the mink was also a good factor, a good candidate for like spreading COVID. There were some patients in North Jutland that was getting a COVID from mink. So when that news came, like only 6 patients, we just actually Denmark government through the prime minister, they just exterminate all the minks, this animal. Uh, in Denmark. Even it was very controversial because we are killing animals. So like the animal lover society, they get angry with Denmark because they're killing this. But this is one of the source of the new. It could be a new COVID variant. So they just exterminate it. But then yeah, it was like a brave movement. Whether it's true or false, I mean, it will make a new variant. It was a brave movement. I cannot say it's true or false, but yeah. And then, yeah, I will talk next about how are they doing in sequence after they sequence it. So after they sequence it, we will see this kind of stretch, a lot of stretch in like maybe 150 sorry, 15 gigabytes, could be 60 gigabytes, any size. And then we just have a lot of stretches, and we need to align it to a reference DNA. This reference DNA, for example, this is a COVID genome, is available for free in NCBI websites. And then this reference DNA has been like a fully sequenced, either it's a de novo or is it Sanger sequencing. And then after it's after some, like for example, result of PCR positive patients has been sequenced like this, we just align it against the reference DNA, and we just match it. Okay, so this belongs to this part of the DNA, and these are unmapped reads, and this is the map reads. We just do that, and sometimes, yeah, this also always happen, unmapped reads. And then we calculate how much of the coverage, is it like 100%, or there are 3% difference with the Wuhan, for example, Wuhan reference DNA. If it's different, then where is the difference? We need to know where is the mutation located. And then, yeah, and the challenge in alignment algorithm, of course, it's computer science related. So bio people have to learn about this computer science algorithm. So it's a, for example, like if this is the reference and this is the read, there can be mismatch, and this is an example of mutation. So it's called a mismatch, and then also it could be an insertion. So like an insertion of 3 letters, three like AAA, and then into the original genome, and it's called also a mutation. It's called insertion. And then also it could be a deletion where one letter is missing, or two letters, five letters, 10 letters, and it just one letter difference can change a lot. I mean, in the effect of this new COVID variants. So yeah, and another challenge is repeats. We can have a lot of repeats like this. So how to, I mean, how to align this reads sequencing result into the reference? That's another challenge. And mapping very large reads to a very large reference database quickly and accurately, it's very challenging. And computer scientists has to think about how to do a better computational complexity, like ON or O log N, better than that, maybe. So we can get the result faster. And this is what we call the metagenomics de novo assembly. So instead of taking samples from like a culture, we just take sample from directly from sorry from, instead of from culture, we just take it from sample from DNA. And this is an example of the sequencing result. As I said before, it's a lot of stretch. And de novo assembly, what a de novo assembly doing is basically changing this stretch into newspaper, back to newspaper. So it's a very difficult job, and this is what a bioinformatician does. And instead of doing only one paper, this is a collection of a lot of newspaper, and we have to do it like this, like a lot of different newspapers from this a lot of stretch. And this is the nanopore sequencing. So this is the size. It's just like this size, and then yeah, it's a USB stick size, and we just need to pipette into this tool. I've tried it once here, and while it's pipetting, they are moving. The sequence are moving in the screen here, or we can also like put the buffer into a file name. So now all information is located in just one small flash disk like this. And so yeah, moving on to the next topic, which is a method that I proposed like more than a year ago, I think in March when the pandemic started. So there was a brain challenge at that time. I proposed this. It was not even in top 10 or something like that. But yeah, it was not accepted at that time. So this is a pandemic detective idea where the idea is to have two detectives, so field detectives, who take samples in different provinces or different places. It doesn't have to be in Indonesia, but in different places. And then this field detective will wander from one area to another, one village to another. And they have a RNA sequencing kit and rapid identification tool, which I developed this during my PhD, this rapid ID tool. And then at each village, they collect 24 samples. They can have the result within less than 10 hours. And then they have to upload files, this result, into the cloud while continuing the journey to the next village, because it's possible that we detect, oh, there is no COVID, but then when we do it more detail in the headquarter, oh, there is this like COVID in this sample. So he's wrong. I mean, this rapid identification is wrong. So we have to do a slower identification tool in the headquarters. So in the headquarters, there are some detectives that are collecting things from the cloud, and then they monitor the uploaded file from different places, different village, and then they make sure that the RNA sequence analysis runs smoothly in the cloud automatically. It's a pipeline, and then they can generate reports and visualization of the map of the progress of COVID-19, and then to communicate with the presidential team, Ministry of Health, policy making, Board of Disaster Management, etc. So that was the idea, where the rapid detection on the field is done in the matter of seconds, minute, after it's finished like in 10 hours process. And it requires minimal bandwidth, rough and dirty identification. And it uses very, very small COVID-19 database, and it's multi-platform. And a thorough detection on the cloud in the headquarters is like going to be detecting whether it's COVID-19, it's not SARS, not MERS, not another virus, and maybe it's no virus at all. And then just to make sure. And then also to make a geographical map of COVID-19 spread. So it should contribute to a global database, and it will be like up-to-date database along with other countries' results. And then hundreds of mutants can be detected there. And then, for example, like in village A, in province A, there were like Italian COVID variants. And then, yeah, this is actually a slide from like a year ago. We don't have alpha variant at that time. We don't have alpha, beta, delta, gamma. So I was introducing like a Wuhan variant at that time. And then there is no COVID. Oh, but then my prediction happened. So of course, virus will mutate fast. And then this has been predicted a year ago that there will be a lot of variants like this. But yeah, that's the function of this pandemic detective integrated system. So we know also when and where a similar COVID-19 variants were seen before. So if we have a new variant, we know where the source is. Like, oh, it come originally from this part of Java Island, from this part of anywhere. That's just an example. And if there is a new mutant, we can update it to the global database of COVID-19 around the world. Otherwise, it's just like, okay, there's a COVID, it's positive, negative, and then it's okay. We don't know whether the vaccine will work or not because we have a new variant that we never know. So this is the importance of sequencing. That's like an example of that is the alpha variant I told you before. That was an example of this kind of system. So this is not my system that is implemented in Denmark, but the similar system. And then after that, we just do some downstream analysis of COVID-19, like genome assembly, virulence ID, drug discovery, vaccine invention, something like that. So I will continue with what's next. But before that, I would like to tell you about some of my doctoral dissertation topics. I hope you're not getting tired. This is, okay, it's around 40 minutes already. So yeah, so in my dissertation, this is my dissertation actually, this is the cover. So the content of that is there is a tool for rapid identification of pathogens using DNA sequencing. So it can be done through a PC, laptop, tablets, mobile phone to identify a pathogen, which can be useful in this kind of time, pandemic time. It was developed like around 8 years ago, these things. And at that time, we're not that massive in sequencing, and we're not kind of predicting this kind of pandemic will happen. And then this is the very first web-based client-side tool to identify bacterial whole genome sequencing at that time. So this is the paper. So the paper was accepted in around 2015, but then it was a work from 2010 and 2013. And secondly, it's a tool for detailed diagnosis using whole genome shotgun metagenomic sequencing. And we are also the first to introduce this concept. So we are sequencing directly from clinical samples. And I was a second author in this case instead of first author, like in the first product of my dissertation. And one of this case that we have solved, I told you in the beginning of the presentation, is this Germany E. coli 0104. It's for outbreak. The cases were very heavily happened in Germany. It also happened in Denmark and Sweden, Netherlands, Poland, Switzerland, UK, and other countries. And there are like 4,000 people infected. 53 died of E. coli disease. And at that time, we find like a source tracking, false source tracking, which we thought it's from a group. It's kind of cucumber from Spain. And then when it's imported to Germany, then the cucumber is thought to be the cause of this outbreak. And then the sequencing guys kind of work. We are kind of start working on it. So we found it's not the source. So the news was false, and then like I think the Germany government was asking for sorry, apologize about that. But then like yeah, that's the importance of source tracking. We call it tracking also in Indonesia. I heard that we start to recognize this trend is called tracing. Yeah, tracing is very important, very important. So we know where it comes from and what's the new mutation. Uh, and then also we were working with a urinary tract infection patients in a hospital in Denmark. There were 25 suspects. So these are some of the selected publications related to outbreak. So yeah, at that time we call ourselves an outbreak detective. And so this is the paper. The first, sorry, the point number 234 in the previous slide was basically this paper, and it already has 332 citations. It's like we are the first to introduce this metagenomics diagnostics. And then there are some, we are also publishing a book here, a book chapter in a Springer book, Applied Genomics of Foodborne Pathogen, if you're interested to read. Yeah, let's start with another example. Now that the pandemic has like a slowdown in many countries in Europe, so our bioinformaticians can start. Uh, okay, so what's next after this pandemic? Of course, the very first thing, very busy bioinformaticians task for bioinformaticians right now is to develop vaccines, of course, and to sequence everything, so COVID related. But yeah, other than that, we can also do a lot because bioinformatics can do many things other than just related to health. For example, like this, my project during my PhD, it was a urinary tract infection detection. So we found that there are 25 samples, and some of them are like they're containing no pathogens, may be healthy. Some contain default urinary tract infection bacteria. The other contain like a newly found urinary tract infection bacteria or fecal UTI. So we can categorize this, and in that case, we can personalize the antibiotics given to this patient. For example, like this patient number 4, there are no pathogens found in the urine sample. Need to find out whether the 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 pathogens are virulent or not by going further into the sequencing analysis. So we found that V583 is kind of dominant or present. So it is dangerous. It is virulent. So we just kind of okay, we have to treat this patient, something like that. But what kind of antibiotic is can be personalized for this person? For example, like in a urine number 3, they are very like resistant to these antibiotics. So we have to give another antibiotics to treat this patient number 3. So yeah, this is also the importance of sequencing. Another project is, for example, we were finding a nosocomial bacteria. For example, if we come to a hospital, of course, the virus is everywhere. So that's why, like, if we remember, like, to throw out your shirt after you're in the hospital, then you just go home and wash it right away. That's because there are a lot of nosocomial bacteria in the hospital. That's why, like, we have to detect what are the nosocomial bacteria in the hospital. For example, we can get it from the wall of the hospital, or sewer is a more efficient one. So in this example, in a Harry hospital, we found like some bacteria, like mostly the one causing a watery diarrhea. But it's normal to find this kind of bacteria in a hospital. And another project is an ancient toilet. We found a 300 years old during excavation in Copenhagen. So we are collaborating with the University of Copenhagen and Copenhagen City Museum when we excavate this toilet, and then we found the feces is still intact, so we just sequence them, and we found this in the feces, this bacteria, which also show that so this maybe people from 300 years ago in Copenhagen, they eat. We can find the health profile, like they eat a lot of high carb, low protein at that time, and they eat a lot of probiotic. Yeah, it's also, we also find that there are a lot of like a trade. Yeah, it confirmed the situation that there were a trade of fruits at that time in Copenhagen, and there are also controls. So this is very important for the sequencing to have a control. And then there are also project number four, a vulture. So vulture scavenge on rotting armadillos died of infectious disease, but they're not infected. So the pangolin or armadillo naturally carries pathogens like leprosy and coronavirus. So the question of this is actually this project is, do pathogens survive their digestive system and emerge at the other end in a viable form? And how come, like the vulture actually spend a large amount of time with their heads inside the sand, eating the rotting carcass without developing sores on their face like a leprosy patient, or even dying of food poisoning? How can they still be healthy? So do vulture build a mechanism to resist such effects? So maybe it's kind of a drug idea. So our project is like this. So the vultures actually wander in the desert and the mountain, go up and down. They eat with their heads inside their victims without developing leprosy. Maybe based on the sequencing result, we hypothesize that the Psychrobacter on their face protect the vulture from the pathogen, and it could be a drug candidate. And when we sequence the gut, their intestines are free from leprosy bacteria and have a low pH. So it seems that the acid in their digestive system may inspire the future drugs. So it's possible. Uh, Another project is a North and South Pole expedition. So we, before I came as a PhD, there has been an expedition around the North and South Pole. Some projects, they take sample from the ocean on the surface and a bit deeper, and then very deep there. So yeah, in Greenland, around this area, also in the South Pole, we found that in the surface part of the ocean, we found like a normal bacteria that we already know, but in the very deep ocean, we found like alien DNA. We never know, but that makes sense. I mean, the fish living in the deep sea, they're like a new species kind of thing. Yeah, so that makes sense. Also, this is the sampling part of that expedition, and we just sequence after that. Another idea is to do a fecal microbiota transplant. So there is an idea where to solve some disease, we can just clean up the gut of a patient and then feed it up with someone else's feces. This is a very like incoming research area that I actually like, because this has solved many cases. Like, for example, Clostridium difficile infection. This has been solving these cases in the US itself. So CDI is like, because this patient is eating a lot of antibiotics, they're already resistant to all antibiotics, and then there is this Clostridium difficile that is, or like, okay, you give me antibiotics, yeah, it doesn't make like us go away. So at that time, the Americans are like saying no, no, no. They're like kind of a bit old-fashioned at that time, because feces is not a solution. Feces is just bleh. So it's not a good idea to use feces as a drug. But finally, they budge, because this drug, this feces of other persons, they're solving a lot of problems here. Like these CDI patients are healed because they are like healed by another person's healthy person's feces. And then also the inflammatory bowel disease has been solved in research only. And then IBS, autisms, and then some genetic disease as well, autoimmune, can be solved with this disease. But in some countries, they're still considered as dirty stuff. So no, it's not ethical to use feces as a solution, as a drug, something like that. Even obesity, this is what interests me a lot. So there has been some example where these obese people, these fat people, they eat not a lot, but they still obese. So it's because their gut intestine bacteria is already full of firmicutes. So there has been experiment on mice. So the obese mice has been like taken the gut has been clean. They put a lean uh, mouse feces inside that, and the 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 mouse is not obese anymore based on this experiment. So it could be used for human, but it's not allowed yet until now. And of course, before I close these things, so the resource needed to do this bioinformatics analysis, of course, metagenomic sequencing data. Without data, what bioinformatics can do, of course. Data is very important. So if you don't have data, then goodbye. Don't do bioinformatics. Also, you need to have a large disk storage and supercomputer. But right now, the era of in the era of cloud computing, everything is like cheaper. If you put it in the cloud, you can build a pipeline in the cloud. It's easier, it's cheaper. So this is not a problem anymore. And data science skills. Of course, bioinformatician is also a data scientist at the same time because they're handling 1 terabyte of data per day or per 2 days, and then they have to finish it. They have to present it to our customer, to our audience, to doctors, to medical practitioners, something like that. That's the resource needed to do bioinformatics on the. So after this pandemic is done, so what's next? Bioinformatics can do a lot of those things, and this is the resource needed. So to close, to sum up, so bioinformatics and cloud technologies have been beneficial in many countries' decision making during this COVID pandemic. Just checking the time, so it's almost finished. So sequence analysis algorithms have solved the outbreak using this a lot of algorithms like gene alignment, and then de novo assembly, gene annotation, gene expression analysis, and many other algorithms, string matchings, etc. And using nanopore sequencing, we can now do DNA sequencing, RNA sequencing from your own kitchen, as easy as that. Doesn't have to be like a big company to do that anymore, but still, with inside your kitchen, you need. It's not that simple still, but it's possible. And using the shotgun metagenomics and bioinformatics and data science, now we can enable finding microbial diversity list of pathogens from a sample, whether it's a blood sample, a saliva sample, feces sample, fecal sample, or a urine sample. And this is a primary key. Bioinformatics is a primary key to discover the cure for genetic diseases, cancer, and then fecal microbiota transplant, and of course this COVID situation. Because like just one more story before I finish. So there is this course in during my PhD. It's called algorithm in bioinformatics. I remember the professors like telling us in an exam. So now we have to build. You have to build a flu vaccine. You have to design a flu vaccine. So now they give me like a step by step. So this is the sequence from the database. This is, and then now calculate the binding affinity, calculate this, calculate that, and then do the reverse complement. So that's what we do. And at the end of the sentence, we say, now it's easy, right, to make a vaccine to make a vaccine for flu, now you can open your own startup company. So that's just anecdote from my previous PhD. So that's all. So that's all from me. This is bioinformatics and the next step. Thanks for the session. I hope there is no one of you getting asleep. Thank you. So, Mr. Tan, I think the floor is yours again.
Okay, thank you, Dr. Danny, for the very insightful talk. Actually, very interesting. Actually, very interesting to see your projects on the microbiome on multiple hosts. And at the end, we try to see how the outcome actually will affect, actually will contribute to the, I would say, the health of human community, the social. Okay, so I think, so now we are in the Q&A session. Okay, so we have this question from the audience. Okay, so this audience asks about how many hours are needed in identifying whether a COVID variant is alpha, beta, or delta by using the current technology. I mean, he or she mean the sequence, the NGS. Yeah.
So the question is how many hours are needed in to
Yes.
identify? Yes. So yeah, it depends on the sequencing technology. Thank you for the anonymous attendee here. But yeah, so it depends. If you're using Illumina, it could take 1-3 days, I think, per samples. But using nanopore sequencing, you can do it like multiple samples at the one time. Oh, it's also possible with Illumina, but still longer. But with, for example, with Illumina MySec, it takes like 4, if I'm not mistaken, like 4 lanes at the same time. But with Nanopore, it can take like 24 maximum if all the sequencing is successful. So it takes less than 1 day in ideal world, but then yeah, people need to sleep also, so it's not really less than 1 day, but 1 day in 24 samples, I think. Yeah, I'm sorry. Before I would like to apologize to mention Mr. So it's Dr. Tan. So
Oh, it's
sorry,
fine, no problem.
I've been in Malaysia, and yeah, this is how we address. Yeah.
Okay, Dr. Danny, thank you for that. Actually, for me, I have a list of questions from your interesting talk, actually, if you don't mind.
Yes.
Okay, maybe I also ask this on behalf of the audiences. Okay, so I group my questions into maybe 3 or 4 groups. Okay, one is in
Yeah.
the general one is about sequencing, and one is about the trend of bioinformatics. Okay, so maybe I will start with, so because throughout the talk, Dr. Danny mentioned about the Illumina, about the nanopore. So actually, maybe our audience, they are not really in the NGS, so they're not really aware of actually the sequencing technology here. So maybe Dr. Danny, can you actually share on how actually nanopore actually advantages to Illumina and vice versa? Because for me personally, previously I understand that when the nanopore actually newly announced in maybe in 2015, but it's not really a favorite for the researcher to use it for the sequencing. But I realized that during this COVID period, a lot of people actually use this nanopore technology for the sequencing. Actually, how there's a shift of trend in this manner? Thank you.
Yes. So yeah, actually, this nanopore sequencing is also also during my defense. They just announced it, kind of thing, in 2013. So yeah, I heard last time the nanopore sequencing. There are some sequencing errors like around 30%, but then it kind of reduced slowly, and they give a lot of thought in making it more. I think on the product they have great eye, and they have like more and more products, um, and then the availability and the fastness, the speed of sequencing is their prominent feature, their advantage. But the sequencing error might be still the hindrance there. But Illumina, of course, has a much lower sequencing error. Although the processing the process of sequencing takes longer than Nanopore, and it cannot do streaming yet. And I heard Illumina is also buying um, some uh, some company. I'm not sure what, I think backed by. I'm not sure, but yeah, all these manufacturers are like competing. There is a race against each other. So they're saying their product is better. There is also Pacific Bioscience. They're also competing. Yeah, so yeah, nanopore is basically more practical because it's a smaller, like a USB stick size, and yeah, more papers are doing sequencing with this nanopore sequencer. That's why I brought up this nanopore sequencing.
Okay, thank you, Dr. Danny. Yes, I agree with that actually. They maybe miss the matter now because of they want to have the fast way to actually to get a genome sequence. That's why actually they really push the platform that actually can do it in a faster manner. Okay. So we start to have some question in from the audience, okay, audiences. So now, okay, this actually recap from what Dr. Danny said from your so-called workshop previously. Now it's that's easy to make flu vaccine and start your own startup company, right? So actually, is it possible for a startup company to do vaccine research and production? What about then? What is your comment about for developing countries like Indonesia?
Yeah, of course, it's not that easy. I mean, this is just an exam question at that time during my PhD. So yeah, designing is easy, but making a startup is not as easy. There are like a lot of talk, elevator pitch with the investors. There's also this uh, when you want to propose a new drug development, you need to go this very lengthy process of this drug development stages like clinical trials, 3 to 1, and then more populations, and then more approvals. Of course, it's not that easy, but designing candidate candidate drugs is easier now with the chemoinformatics and some machine learning algorithms. What is that answering the question? I hope. But the developing countries is the same. Developing countries, they all have the same way of drug development phases. So
Okay, thank you for that. So the next question here actually is on the maybe on doctor's previous work on the classification, I mean the identification of the bacteria. So what method do you use? Sorry, what method did what you use, I think, pattern matching of the sequence and classify the sequence and what is the accuracy? Okay, actually this is also one of my question where actually what is the potential markers or maybe databases that doctor use for the identification purpose and the classification purpose? Is there any like, application of machine learning as well?
okay, in that case, yeah, we need to use this conserved region of the the what do you call it, the target itself. Like for example, for bacteria, we use 16S. For example, 16S is a part of the genome. So the genome is that long, and then the 16S is a few hundred letters. Uh, that is always like constant, almost constant, almost 100% constant, because the other part sometimes it's mutating easily, but this part is constant. Then we find that constant part and use it as a marker. And for virus, it's a bit difficult because it keeps changing in all places. But there are still some conserved regions that we can take as a marker in this case. So, yeah, that's the prior knowledge that we need to have before we design markers.
Okay. Uh, But I get to know that for 16S, maybe for certain complex like MTBC, I mean like tuberculosis complex, their 16S are very conserved. So actually means that the tool that or the kit that doctor actually shared with us just now is that like certain up the classification only can up to certain level or can
Yeah.
up to really deep down to the subspecies level or species level.
Oh yeah, okay. You're right. So okay, that's the question about. So the yeah, that one is for the dirty, rough and dirty identification. So we can like have a sequencing file and then like detect it faster. But then when we want to do detail typing or maybe de novo assembly, genome alignment, or maybe finding a mutation, somatic mutation, I don't know, or something like that, we really need to go into everything, all. And then we have, for example, like we have to align first. If it's something new, we have to do de novo assembly. And yeah, and then from there, we can detect where the insertion or deletions or the mismatch happens, and then we can take it from there as a snip is
Okay,
yeah,
okay.
snip. Yeah, I don't know how to explain. Yeah, it's the difference. I mean, yes.
Okay, okay. So we reference to the question for the audience. So he or she was asking about the method. So means that for this kit, we are using the doctor is using the similarity based approach. Similarity based, right?
Similarity, yes, of course. Yeah, we
Okay.
find the difference. Yeah.
Okay. Thank you. So we had a next question. So if you don't mind, doctor, sorry, because of your interesting area here actually. Okay, I want to ask technically from AI side, how many data do we need to proceed genome sequencing model? How do we extract the feature to determine the next genome, which algorithm? Uh, Sorry, Mr. or Miss Asril Jarin, can you actually elaborate more on your question? Or doctor, do you get the question from this audience?
I think I might get it. So
Oh, okay, okay. Good.
so this thank you for the question. So I think this is from a computer science perspective instead of from bio perspective. Yes, in this case, if you want to do a genome sequence to proceed with a genome sequencing model, that's not the AI that we want to do, like by doing this feature extraction or the number of data. So genome sequencing actually not yet related to that. AI is something else. In bioinformatics, we just kind of uh, mostly we are doing a string matching, and we already have tools everywhere. Like it's called PWA. There is a bow tie. There are many other genome alignment software there, and you can develop your own algorithm. It's called string matching algorithm. So we are our everyday's work as a bioinformaticians are mostly uh, doing this string matching. So if, and AI is useful usually for calculating this when we want to find this binding affinity kind of thing. So when we want to find a candidate drugs from available active compound, something like that. And yeah, there are many places, but not in DNA sequencing analysis. Yeah.
Okay. So we have a lot of questions here. I'm sorry, but I have to select a few. Okay. Maybe about this. Okay. Talking about data such as because in bioinformatics we are actually dealing with databases, right? So there's
Yeah.
one audience here talking about data that such as GenBank, UniPro, KGG, etc. that
Yes.
frequently used in bioinformatics research. Do Indonesia or each country need to build their own database? Any comment on this, Dr. Dani?
Yeah, in my opinion, we need to unify all database from every country like into one server. And right now, the current server is NCBI. So yeah, we don't have any other servers like all other servers. Sorry, I mean all other server are like submitting to that NCBI server. So yeah, right now we only have one, and we should not have more than one. Uh, and that was actually I still remember if it's okay also I have a little bit story during my PhD when we propose virus database sharing in 2011 I guess or 2010. So we got like a lot of complaint no like not complaint like a counter answer from the people from CDC. I think at that time they're our not CDC, yeah, CDC or WHO. I don't remember. They were like giving answer, no, but it won't work. Your virus database sharing, because there are some countries that are rejecting the idea of this database sharing. But yeah, that's our homework to make it like available to make everybody want to share their virus database so that everybody can access it. Everybody can have the same what do you call it? Have the same opportunity to invent a vaccine or drug candidates.
Okay. So actually, I agree with Dr. Dani about the collaborative and the unified kind of data because even like for GenBank in which hosted in US and maybe EBI hosted in Europe and the DDBJ hosted in Japan, they also frequently actually exchange their data for the unified purpose, for collaborative purpose, as what Dr. Dani mentioned just now. Okay, so actually, Dr. Dani, we still have the time for Q&A. Actually, are you okay if we have a few more questions? Okay,
Of
very thank
course.
you for that. Okay. Okay, so we have one question here. Dr. Dani, could your bioinformatics result to be used to prevent and control in Indonesia, country with huge population during the pandemic outbreak? Okay, so I mean, I think basically what's the audience here means that how I mean maybe in general, okay, how's actually bioinformatics, the output from bioinformatics can use for the prevention and control from any kind of outbreak. Okay, maybe specifically, maybe doctor can bring down to the case for COVID-19. Thank you.
Yes. So the most important thing is sequencing and sequencing and sequencing. That's how to solve this. I mean, so we tracing is very important, very important. I talk in many I don't know webinars or something like something like this. A lot of time sequencing and sequencing is always the and tracing related to sequencing is very important. So when someone is infected, uh, for example, like PCR positive, don't stop there. Take the sample, sequence it, and then find it out, and then make this person like what do you call like educated. Like okay, you have to close your circle. So who did you have contact with in the last few days? Also, like for example, in Denmark, we have this Coronavirus Meter application where everybody install it, and if you have been in contact within 5 minutes with the person that has been like positive PCR, and then the other person will get like a okay alarm that okay you need to get a PCR test, something like that. Tracing is very important. For example, the other way to end this pandemic, just like what we did in Europe here, is of course a lot of tests. And in Denmark, especially, it is like very successful because the PCR is for free. PCR testing in Denmark is for free, and people have to do that if they want to go, if they want to enter a shop or something like that, or they want to cut their hair, they want to go to a boutique, they have to show the proof of PCR negative, for example. So that's how we reduce the number of infection in Denmark and sequencing after it's positive, of course.
Yeah, I mean, maybe I can add something to what Dr. Danny actually mentioned as how actually the involvement in bioinformatics can actually alter or actually suggest a new policy for the national level to tackle certain issues like the outbreak now.
Yeah.
Okay, and maybe one of the clearer actually application of bioinformatics is the detection of the variants, the VOC. That's where actually we managed to get the alpha, beta, gamma, and delta, and all the variants of interest. Okay, that's actually the direct application that we can see clearly from the bioinformatics. Okay, okay, thank you, Dr. Danny. Oh, here we have very interesting question and very good question here. Okay, Dr. Danny, how do you deal with the data that contains personal or individual DNA genome sequence information when you need to share the data to other researchers? So here is touch on the data protection matter, I think. Okay, Dr. Danny?
Yes, of course. It's we need to have this NDA agreement with the patient when we are going to use it as for research. It's already been um, like a normal situation in here. So if research want to use our data, for example, anything, not only for COVID, like for cancer, everything. Uh, if you want to go to this treatment, please sign this NDA that this will be used. Maybe there is also like anonymity. Is also, if you want to be anonymous, just say anonymous. Not, it's been part of this policy in I think all countries in Europe if they want to do this research, and the US as well.
Okay. We have one question here. Okay, bioinformatics seems to be the future medical trend. Okay, is there any use of bioinformatics for cancer treatment or prevention? This is actually related to cancer. Okay, I mean the general application of bioinformatics in the medical treatment. I've heard that cancer is genetic, so maybe bioinformatics can be used for the treatment or prevention of someone who have a cancer risk. Are there any country developing this technology? And for Indonesia, are we still far from being able to implement this thing? Dr. Danny?
Um,
Mm.
yeah, as long as sequencing is not popular yet, then I don't think it's going to be that massive. Of course, yeah. So, for example, like in Sweden and in Denmark, so all the patients who wants to get tested for cancer, they they were taken samples, and then there were some bioinformatics analysis behind that, where they detect the mutation, the typing actually. For example, like in breast cancer, for example, uh, they sequence the sample of the tumor of some early stage breast cancer a patient in that case, and then we do some kind of identification whether this is like estrogen uh, ER. I don't remember what's it, like ER positive or HER2 positive, or both, or none of them is something else. So like triple. But then like if it's ER positive estrogen, if the breast cancer is caused by too much estrogen hormone, then just suppress the hormone. If the cancer is caused by a HER2 hormone, then just suppress the HER2 hormone. There are some treatment for that. If both, then give both suppression. And for others, then there is other solution. That's an example of how we use sequencing to solve this breast cancer patients cases in Sweden and Denmark, as far as I know. Yeah.
Okay, so actually they are actually people using these bioinformatics for the treatment. I mean, for actually this cancer purpose. Actually, in even in Malaysia, okay, there are actually some of the uh, independent agencies also actually actively engaging with bioinformatics to cater for this cancer medication or something like that. Okay, so actually it's active ongoing, but as what Dr. Dani mentioned, this actually made not possible if the sequence is not famous yet in certain countries, right? Mm.
Yeah.
Okay, because no means that down to earth, I mean down to the reality means that we still need the data. Okay, that's the key for bioinformatics here. Okay, so maybe I have another question for Dr. Danny, if you don't mind. Okay, about the Mings just now. So
Yep.
just now, Dr. Danny mentioned about the Mings as the there's a issue with the SARS-CoV in Mings, something like that.
Mm.
So may I know, can you share if anything that he came across, doctor, that actually reported for any animal that can cause zoonotic infection for this SARS-CoV-2,
There
other
has
than the
been,
Ming, sorry, yeah.
yeah. So I think the first mink is the first animal to be known infected by COVID, uh, and then there was also this feet of ocean bird, one of the
Oh,
ocean bird. I think seagulls. Yeah, seagulls. They
seagull.
found a trace of COVID in the feet of the seagull, and then from there are like a lot of animals. I just heard recently in Indonesia, in one of these zoos in Ragunan in Jakarta, there are two. Is it lion or yeah, or yeah, that has been infected by COVID. So there's no wonder. So it can infect
Mm,
animals, and it can transfer from animal to human. It is possible. You know. So that's what the Danish government did when they heard that Ming is the source of this 6 or 8 patient, less than 10 patients getting COVID in a Ming as a Ming owners, I think, or Ming workers in a Ming farm. So they just treat the patient, and this sequence is quite new. So at that time, they think the vaccine won't work for this kind of patient, so they just kill the mink so there won't be a new variant of COVID. They kill all the minks in Denmark. But yeah, yeah, it's very controversial at that time. But yeah, it is possible.
I see. Means that it actually indirectly means that the COVID cases actually is more severe than what we expect because you're so infecting human, I would say. I mean, sorry, infecting other animals. Because all this one, maybe the research has been focused too much on the bat and pangolin as the potential source for this
Oh
coronavirus.
yeah,
Okay.
yeah. Then
Hmm.
Ming is the second there. You're right.
Hmm. Okay. Maybe one last question as a general as a okay last question. Okay, so since Dr. Danny actually, if I didn't hear wrong, Dr. Danny actually originally from computer science background.
Yes.
Okay, and then later part only Dr. joined as a bioinformatician, I mean in this area. So can Dr. share actually what are the knowledge or the skills they require because since computer science actually quite close to bioinformatics, but of course there are some differences there. So actually what can we expect or what should we prepare ourselves to engage in bioinformatics?
Okay.
Okay,
So
thank
for a
you.
computer scientist, yeah. So for computer scientist, we need to be very strong in algorithm, for example, like string matching algorithms, and then how to make it faster, mostly in a computational what do you call it, complexity. So how to make it from on square to on, and then to o log n, and then yeah, always make it faster and faster. And then also, when you want to learn bioinformatics, you really have to learn about biology. You have to understand how biology works. For example, like when I started as a bioinformatician, I learned a lot from the biologists in the department. A lot how the DNA works, how the DNA replicates, and then they from DNA to RNA to protein to metabolite, something like that. So it's not as simple as you you know the algorithms and that's all. And that very important skill is not only data science but also this how do you work with parallel processing. It's very important. And how do you work with big data? Of course, as a data scientist, you really need to know how to analyze 1 terabyte of data in the matter of minutes. That's a very computer science skill. Yeah.
Okay, thank you for that, Dr. Danny. Actually, we just received one question. Doctor want to accept the questions. Okay, can you teach us, or we can, Doctor, roughly brief on how we detect the new variants? Okay, what do the ASCII sequence on the slide mean? The FASTQ file format, I believe. Is the ASCII code such a representation of the feature or matrix in the sample?
The ASCII in the sequencing is actually the quality of the reading of the sequencer machine. So when we read like one stretch like 300 base pairs or 300 letters in this case, we for each letter we have like probability or the quality of reading more precisely. So this letter has a quality of 62. It's the ASCII letter can be translated. For example, A is 64 or 62, I don't remember. And then yeah, B is 65, 64, 65. So and then this is lower, this is higher. That's also the challenge in computer science, how to deal with different qualities per base pair or per letter. So that's that ASCII part means is the quality of the reading.
Okay, so I think that's all. Okay. So thank you, actually, Dr. Danny, for all the questions. We can't thank you enough, actually, for answering, okay, the question from the audience, and of course for your interesting talk. So I also like to talk. I also like to thank the audiences, okay, for the participation. So again, thank you, Dr. Danny. Sorry. Okay, I would like to pass the floor to Master of Ceremony. Okay, thanks, Dr. Danny.
Thank you.
Okay.
Tan.
Thank you, Mr. Saputra, PhD, and Dr. Tan for this amazing session. It was very amazing for getting your insightful knowledge. And now we would like to kindly invite both of you to have a photo session. All right, so we would like to count 1, 2, 3. 1, 2, 3. Is it okay if we ask you to take a photo for one more time? And please give the sweetest smile for all of us here. All right. Okay, thank you. So we would like to count 1, 2, 3. Thank you. Okay, thank you, Mr. Saputra, PhD, and Dr. Tan for this amazing session. Now, ladies and gentlemen, we're going to have a short break. And if we would like to remind all of you, if you would like to rewatch the whole session of the NIT ICoICT 2021, please refer to our YouTube channel, Telkom University, or School of Computing, Telkom University. And also, we would like to remind all of you that don't forget that tomorrow we're going to have the third, or I do apologize, this afternoon, this evening, we're going to announce the award for the Best Paper Award of the NIT ICoICT 2021. And now let's have a short break, and please do kindly refer and return to the room at 10:15.
A wondrous civilization. Disposed to tranquility. with scenic landscapes bordered by the sea. Here, mythologies become principle. And religions create the cornerstones of life. It is contained in the arts and crafts, where celestial ceremonies become part of popular culture. Experiences you can take home. Here, art preservation is a tourist attraction. History is a living monument. Even shopping is meaningful. See world famous or the excitement of exploring nature. With endless stories to tell and never-ending feasts. Never-ending possibilities to get more out of life and journeys. Yogyakarta. Wonderful.
Nowadays, the need to have some conveniences, amenities, and secure data services have become an important part to every business owner. Also, they want to have the system to be in a place that's affordable and accessible for them. Cloud computing appears as the solution that meets the challenge of digital era and can be adapted as per industrial needs. Through digital platform like cloud provider, you don't have to supervise or buy some data storage machines and servers, which you require a regular maintenance. Moreover, it will be a big advantage for your company to have a cloud system that has its server base here in Indonesia. Introducing Flow Cloud, the best cloud services solution by Indonesia for Indonesia. We are the solution for your company in need of IT services on cloud computing, with a global standard on capabilities and technologies. With the development plan for national digital infrastructure as the accelerator of digital hub in Indonesia, Flow Cloud supports the industry to develop digital networks and platforms in order to become the digital industry 4.0. This is the answer for some customer experience to you. A startup company which focuses on serving online payment uses cloud computing to build an app that can help the small restaurant to do a transaction with their customer by using a smartphone. Many enterprises use cloud computing to build a better infrastructure to fulfill what market needs. In the government sector, cloud computing takes a major role in big data analytics as a part of decision-making to create policy. Flow Cloud has its servers scattered around Indonesia with the best quality, the highest performance security, and the fastest connection. Therefore, you can have the best cloud computing for your business growth with minimum expenses. Flow Cloud supports your business to grow first. After that, let's accelerate together with us.